In this presentation, I will review a grant proposal by Professor Heather Mortiboys titled Secondary Mitochondrial DNA Damage in Parkinson's Disease, a Target for New Treatment Strategies. So, to summarize the background and rationale for this proposal, mitochondrial dysfunction has been well documented in sporadic Parkinson's disease and in toxin-induced cases, such as those related to MPTP. It involves respiratory chain dysfunction and mitochondrial DNA deletion, which are increased in the substantia nigra in sporadic cases. However, mitochondrial DNA damage in familial Parkinson's disease has not been fully investigated. Parkin and PINK1 are genes involved in mitochondrial DNA maintenance, and their dysfunction has been associated with the dysfunction of mitochondrial functionality. Mutations of DNA polymerase gamma, which is one of the only polymerases for DNA, are associated with Parkinsonism. Evidence from the MitoPark mouse studies has further substantiated the mitochondrial dysfunction in Parkinson's. The proposal here aims to elucidate the role of mitochondrial DNA damage in Parkin and LRRK2 mutant Parkinson's disease, and to assess if mitochondrial DNA damage replacement by DNA polymerase gamma overexpression is a rational therapeutic strategy. The crux of the experiment is hybrid generation, which are cytoplasmic hybrids that contain mitochondrial DNA of one cell in a new nuclear background. Deep sequencing will be performed on primary Parkin and LRRK2 mutant fibroblasts, as well as the hybrids formed, to see if wild-type Parkin or LRRK2 nuclei can rescue mitochondrial dysfunction in a cytoplasm with mitochondrial DNA from Parkin or LRRK2 mutant cells. Quantitative PCR as well as Western blotting will be used to measure transcription levels of certain genes such as TFAM. Mitochondria will be further tested for function and morphology. Polymerase gamma DNA will be overexpressed transiently in senescence-prone fibroblasts and in the SH-SY5Y cell line hybrids with mitochondrial DNA from Parkin mutants. The importance of this project is supported by Parkinson UK's priority of understanding why neuronal cells die. These experiments validate if gene therapy targeting mitochondrial function would be a viable option in familial Parkinson's disease. This and the enhanced understanding of pathogenesis stemming from this project would further improve the potential for future therapeutics. In addition, mitochondrial DNA damage is seen in other neurodegenerative disorders, such as motor neuron disease, and any findings will be useful transdiagnostically. The main benefactors for this research include scientists and patients. At the time of submission in 2010 by Professor Mortiboys, mitochondrial dysfunction in sporadic Parkinson's disease was topical, yet the importance in familial disease had not yet been investigated. In addition, studying mitochondrial DNA in LRRK2 was novel. None of the hybrid studies performed at the time had investigated mitochondrial DNA integrity in either the primary patient cells or the hybrid clones, but focused on the downstream mitochondrial dysfunction. Also, this study is the first to investigate if mitochondrial DNA replacement is a rational therapeutic strategy. Prior to submission of the proposal, the ethics approval was obtained by submission to the local committee. Dr. Mortiboys also has a background in biochemistry and has an excellent track record when it comes to research in Parkinson's disease, as she had worked for more than 9 years in the field. She also has experience with mitochondrial dysfunction specifically. Some key articles prior to the proposal are seen on the right. Dr. Mortiboys has also built an excellent network of collaborators for this project, such as Prof. Rattray and Prof. Azuz. These collaborators increase confidence in the feasibility of completing the project within the time frame of 36 months as proposed. As certain components of the project are time-consuming, Dr. Mortiboys has requested salary for a part-time technician. When it comes to strengths, deep sequencing boasts determination of heteroplasmy down to the level of 5%. In addition, this study uses human tissue from Parkinson's patients. Another positive feature is the intent to disseminate findings via publications and presentations in relevant areas. In terms of weaknesses, this study will be poorly powered given the small sample size. In addition, fibroblast tissue is non-neuronal. Mitochondrial DNA deletions and maintenance may not have a major role in peripheral proliferating tissue, such as fibroblasts, as it does in post-mitotic neurons. Heteroplasmy also means the introduction of variability. Another factor which widens heterogeneity is that transferring mitochondrial DNA also introduces the other proteins from the cytoplasm. A concern is that hybrids containing mitochondrial DNA from patients with alpha-synuclein mutation have previously shown no difference in mitochondrial activity between control and patient hybrids. However, mitochondrial function was not determined in primary patient tissue before hybrid generation in that study. One more concern is that polymerase gamma overexpression has not increased mitochondrial DNA levels or mitochondrial function in previous studies. However, these studies only use cells with normal mitochondrial DNA levels and mitochondrial function. In conclusion, this is an interesting, original, feasible, and important study with a good level of work leading up and submitted by an applicant who is an expert in the area. It may pave the way for future discoveries in therapeutics. Based on my review, I would rate it 8 out of 10 on the NMRC scale.